So I will start up this presentation on blood biomarkers, and I will cover only A beta and tau, and Henrik will continue with NFL and some other biomarkers. So to start from the beginning, why do we need biomarkers in blood? We have already PET scans and MRIs and lumbar puncture tests. So I think the answer to that is well known. We soon also have the drugs, of course, we hope. So PET scans, they are not generally available, only in very specialized research centers. They are expensive. I've heard quite a number of different prices. Today, I think I heard $10,000, which may be too high, of course, but still. CSF tests, they are well validated, but first of all, it's a bit complicated to do. And second, there may be side effects in the form of post-lumbar puncture headache. So I think it would be great with this type of tests for ATN, pathophysiology, or amyloid, tau, and neurodegeneration also in blood samples. And that has recently become more hopeful, I think. So how did it start? If we start with plasma A beta, there has been measurements in CSF since long, and that was ELISA methods, and these ELISA assays were also applied to plasma samples. Here is an overview, like a meta-analysis of some early studies on A beta 42 and 14 plasma using ELISA, and as you see, there's really not much of a change using this type of method. So what could be the reason for this? I guess there are several reasons, but one reason you see here, just some thoughts on measurements in blood samples. So if we start with CSF, the concentration of A beta 42 is around 800 picograms per milliliter in healthy people or something like that. So how much is 800 picograms per milliliter? That's very difficult to think of, but if you translate this just by simple maths to something else, so it's the same as 800 nanograms per liter, or 800 micrograms per cubic meter, or 800 milligrams per 1,000 cubic meters. And that's the same size as an Olympic swimming pool. And a sugar cube is 4 grams. So if you take 1/5 of a sugar cube and throw it into a swimming pool, then you have a concentration of A beta 42 in CSF. If you then go to blood or plasma, it's only 20. So it's 1/200th piece of a sugar cube in a swimming pool. And then you have a big problem, and that's all the other proteins, which is around 300 micrograms per milliliter, meaning 50 tons of other proteins like albumin, IgGs, and all the major plasma proteins. So you have to have really high precision and sensitive methods. And that has recently been developed. So one method is called single molecule array, or SIMOA. This is a very early paper on this from 2011. So single molecule array is beads, magnetic beads that are coated with the antibody, and then you have an immunocomplex with, for example, A beta then, together with a labeled antibody on the top of the beads, and you have quite a high number of beads, and each bead will in the end fit in a small microwell, so that you do a digital quantification of the positive beads who have captured A beta and the negative beads who have not captured A beta. And then you can come down in analytical sensitivity to extraordinary low levels, in this case 0.03 picograms per mil, which is close to nothing. Another example is mass spectrometer methods. This is one paper from 2014. So it's immunoprecipitation, where you use an antibody to capture A beta followed by mass spec. And you can do two variants of mass spec, either a variant called MALDI-TOF that you see here. There you see all the peaks for A beta 42 to the right, which is very small, A beta 40 to the second right, which is a major A beta form in both cerebrospinal fluid and plasma, and then quite a number of other smaller species of A beta. And you can also quantify A beta 42 and 40 by single reaction monitoring. And how has this been working? These two methods, CIMOA and IP mass spec. Here is one example of a paper on CIMOA, so plasma A beta 42 and 40 in a large study, the Biofinder cohort from Sweden. So more than 700 people with AD, MCI, subjective cognitive decline, and also healthy controls. So here you see that there is a correlation between, first of all, plasma A beta and CSF A beta. It's not very tight at all, as you see, but it's significant. You also have an association with amyloid PET. So more amyloid in the brain, lower A beta 42 to 40 ratio in plasma. And last, you have a slight but significant reduction in the ratio in plasma in AD and also in MCI cases. So this is promising, but not very useful maybe in the clinic. Then came this paper. This is by Randy Bateman and Fagan and colleagues at WashU. So this is an IPMS method where the whole procedure is started by treating plasma with protease inhibitors to avoid degradation and also guanidine extraction of A beta to release peptides from binding to plasma proteins, and then thermo orbitrap mass spec in the end. And suddenly the situation looks much more promising. This is a slight but significant reduction in A beta 42 to 40 ratio in amyloid positive cases by PET, ending up with an AUC as high as 0.89, which is very good, of course. Another paper came the year after. This is a Japanese group or a company together with Colin Masters group in Australia. So they used instead MALDI-TOF. And in addition to 1 to 40 and 42, they also measured a slightly extended A beta variant starting at position -3. So you could call it A beta -3 to 40 or APP 669 to 711. And here you see the data, and they express this ratio in the other way around. So they have 40 to 42 and so on. And they also combine these two peptides with 42 in what they call a composite biomarker, and ends up with, again, really high AUC values to identify individuals having brain amyloidosis. So very promising results. But I mean, there has been quite a number of other papers, but an interesting and important thing I think is shown very directly in this paper, and this is a comparison between plasma and CSF A beta 42 to 40 ratio. What you see here to the right, that the CSF difference is much more clear than the plasma difference. So in plasma, it's a 10% reduction in the ratio, while in CSF, it's a 43% reduction in this paper. So still, it looks like CSF measurements perform better and has a greater separation than plasma measurements. but we have to see in the future how it will end. You can also measure these peptides by fully automated instruments, for example, the Roche Elexys instrument. This is again the biofinder cohort. Here you see plasma A-beta 42 by itself, a clear reduction in AD dementia cases, but also to some degree in amyloid positive MCI and cognitively unimpaired. In this study, there was also a reduction in A-beta 40 actually, but if you make a ratio, you see that the ratio is lower again in amyloid positive cases and ending up with AUCs above 0.8, which is of course good, and also that it's a fully automated instrument. How about total tau? We know that total tau or T-tau works very well in cerebrospinal fluid. It's very similar and correlates tightly with phospho tau levels, for example. Here is plasma tau using Simoa, but then it doesn't look so good. So there's only a very slight increase in AD, and the reason is that there is actually no correlation between plasma and CSF levels for total tau. Probably because only a small fraction of tau you measure in plasma comes from the brain. And this has been also shown in quite a number of studies. This is the ADNI cohort, large number of AD, MCI, and controls, but really close to complete overlap between groups. So unless a unique combination of antibodies can detect some brain-specific tau fragment, then we can't use plasma T-tau as an Alzheimer disease biomarker. How about phospho tau then? This is the very first paper by a Japanese group on phospho tau in plasma. So they used the Quanterix total tau assay, and then they changed one antibody and of course the calibrator to phospho tau, and examined a small cohort of AD dementia cases and controls. And here you see that there's really a clear increase in AD, which is very promising. Not all samples could be measured, but the good thing is also that in contrast to total tau, there's a very clear correlation between CSF and plasma levels of phospho tau 181. So the first study with really promising results. And then came this study by the group at the Mayo Clinic, Michel Mielke and Ron Peterson, in collaboration with Lilly Research Labs. So this is a mesoscale or MSD method for phospho tau 181 and a large cohort of patients in the AD continuum. And here you see the data for phospho tau 181 using the MSD method. So again, a clear increase in AD and also in MCI cases that are amyloid PET positive, and a good thing, a correlation with tau PET. So more tau pathology, higher phospho tau levels in blood.
My speaker.
And this study was used then in two very large clinical cohorts, one from UCSF, and here you see also a number of other neurodegenerative disorders, not only AD, where there is a clear increase, but also CBS, PSP, FTD, and PPA with fully normal levels of phospho tau 181 in plasma, which is promising. This is the Biofinder study again, just a small extract of the results. Here you see the correlation with tau PET. So tau PET negative patients, they have low phospho tau levels, and then you see increasing levels with increasing amounts of tau pathology in the brain. We in Gothenburg also started to work on Phospho Tau assay, but used Simoa and got that to work. And one of the first things we did was to check the correlation between plasma and serum and CSF, and that looked quite good. So there is a rather tight correlation between plasma and CSF levels, and then examined a number of clinical cohorts. Here is one. This is the triad cohort from Montreal. So Pedro Rosa Neto, very similar results to the other assays, clear increase in AD and amyloid positive MCI cases. Here you see the biofinder data, very similar, and again, also normal levels in Parkinson's disease, PSP, and other dementias. So very promising data for Phospho Tau 181. This is just a separate study we did with a group in Italy. So quite a large number of FTLD spectrum patients, and then AD patients and healthy controls. And again, the important finding here was a clear increase in AD and then normal levels in the other disorders, that you can differentiate the FTLD cases from AD with quite high accuracy. So this was all on Phospho Tau 181. And here you see data on the very early phase of AD. So this is individuals, cognitively unimpaired people who have either amyloid pathology by amyloid PET, that's the A-beta positive group to the right, or amyloid negative cases to the left, but all are tau PET negative. So you can't detect any tau pathology by tau PET, but still you see that in the amyloid positive cases, you have an increase in Phospho Tau 181 in plasma, also with a rather high AUC value. How early can you go then? One way to test this is in FAD cases where you know the expected year of onset. So this is a collaboration with a UCL group in London. So this is FAD cases, symptomatic and asymptomatic mutation carriers, but also non-carriers, family members. And here you see that symptomatic cases, they have clearly high Phospho Tau levels in blood, but also pre-symptomatic cases. And if you plot the Phospho Tau levels in relation to the expected year of onset, you see that these curves start to diverge, something like 16 years before the expected year of onset, so very early in the course of AD. Then there are actually other Phospho Tau epitopes that can be measured in plasma. This is also a Lilly research lab method for Phospho Tau 217 in the biofinder cohort, and to make a very large study very short, you see that if there's really a clear increase in Phospho Tau 217 in AD, and you can differentiate AD from all other disorders studied in this paper with an AUC of 0.96, so really high performance to identify AD, really promising. How about if you go to the other end of the spectrum, to neuropathologically confirmed cases? So this is from the same paper. This is the Arizona neuropathology cohort, and the plasma samples were taken around 3 years before death and then measured using the Lilly 217 assay. And here you can see two things. One is that, again, similar to for tau PET, phospho tau concentrations in blood increase with increasing amounts of tau pathology in the brain, or tangle density score, and the AUC to differentiate AD cases from non-AD cases is as high as 0.89. This is another neuropathology cohort we did using our 181 assay, and here plasma samples were taken 8, 4, and 2 years before death. And here you see the results, so very clear increase in AD already 8 years before, and normal levels in non-AD cases, and the AUC ends up at 0.97. So again, very clear data supporting that phospho tau in blood is specific for AD type tau pathology. And then it's not enough with 181 and 217. You can also measure, for example, 231 in plasma. So another amino acid that can be phosphorylated. This is a paper by Nick Ashton in our group from this year. First of all, we showed by immunoprecipitation and mass spec that this assay really measures phospho tau 231 specifically, and here you see data from the triad cohort, so very similar to the other phospho tau biomarkers, high AUCs to identify amyloid positive MCI cases, and of course AD cases, and some neuropathology data also with great performance to separate the AD dementia cases from non-AD cases in samples taken before death, of course. Since there are 3 different phospho tau epitopes, there has been a discussion on whether they perform differently to some degree. So is 217 better than 181, or is 231 earlier than 217, and so forth. Today, they haven't actually been studied in the same cohort, or at least not published today. But here is a CSF study where we employed Simoa assays for 181, 217, and 231 on the same cohort. And here you see the correlation to the left between 181 and 217, and then 181 versus 231, and then 217 versus 231. And it's clear that they all correlate very tightly and perform very similarly, at least in cerebrospinal fluid, and we have to learn whether it's possible to identify any type of difference between these phospho tau epitopes in plasma samples. We have to await these studies. But to sum up, I do believe that we have quite good biomarkers to monitor amyloid and tau pathology in the brain using blood biomarkers. Thank you, and I leave the word to Henrik.
All right. Actually, just before we, let's see, that's your last slide there, right? So yep. If you want to pull that, Henrik, you're going to put on different slides, if I recall. So let me just actually ask, before we switch over to Henrik, Kaj, I just wanted to ask you a clarifying question. So we have a very broad and interdisciplinary audience of people here from many different fields and many different backgrounds. So there was obviously a huge amount of data and work that you presented. So I just want to ask you 2 real, very simple questions, if you could sort of just define them for a broader audience.
Okay.
What does it mean for tau to be phosphorylated? What would be the difference between phosphorylated tau and non-phosphorylated? And the second question is, could you try to provide sort of a layman's understanding of what do all these numbers, p-tau 171 versus 231 versus so forth, what are those, what should we be thinking about when we think about these different numbers? What are they referring to? So
yes, I will try to briefly. Yeah, so tau is a normal brain protein. It's located in the neurons, and it binds to the microtubules. So its function, at least one of the function, is to stabilize the microtubules. by binding to them. Sometimes I try to think of it like if you have a Lego tower, and when it's really high, then it becomes unstable. But if you place tape along the sides, then it's stable again. That's tau on the microtubules, so that it doesn't sway too much and fall apart. And if it becomes phosphorylated, then it's, now something happened. Then it starts to...
I don't know why that...
I didn't do it. No?
No? Okay.
I think it comes from Millie.
Millie.
Millie's signing off. Something has gone wrong, so I'm signing out.
Okay.
Okay, bye.
Okay, you want to try again?
Yeah, when tau becomes phosphorylated, it cannot bind to the microtubules, so they become unstable. And another thing is that for some reason, tau gets a tendency to aggregate and form tangles that you find in Alzheimer's disease. So tau is a long protein. It's 440 amino acid, or 41 actually. And the numbers, they just refer to the amino acid that is phosphorylated. So amino acid number 181, that's phospho tau 181, or 217 or 231. We don't know really what it means, the difference between these different phosphorylations. So tau is phosphorylated at many different positions in, so there are many more phosphorylations that I didn't mention because they are not regularly measured in CSF or plasma. We don't know the difference. We know one thing is that the same, to phosphorylate tau, you need enzymes called kinases. And these three, at least, the phosphosites 181, 217, and 231, they are phosphorylated by the same enzymes.
The same what?
The same enzymes. So yeah, enzymes. So maybe that indicates or suggests that there shouldn't be too much of differences across these phosphosites. I think, at least, since they are processed in the same way in the brain.
Great. Okay, that's very helpful for those of us not from a neurochemistry background, but so the basic idea is that there are some hundreds of these amino acids in the tau, and these numbers refer to sort of where they are from sort of left to right, and then as these various amino acids within the tau become phosphorylated, that's part of the Alzheimer process. That's part of what causes the tau to break down and for the structure of the neurons to begin to collapse. Is that right?
Yeah, exactly. And tau to aggregate.
That's a wonderful sort of lay explanation that helps provide some context. Let me turn over to Henrik. Do you want to share your screen and give us part B?
Absolutely. And I will also talk about some structural proteins. And I will show you a picture that could help clarify a little bit also what we try to do in the lab here in Gothenburg. But I wanted to start with this slide, because in the upper part here, you see our lab, our people who will receive samples from you. It's a bit old, this lab. You can see Kaj looks a bit younger and I look a bit younger too, right here. But we need to take a new photo, Kaj. And then we have new, we really need to do that. Then we have new lab members also. And Mark knows Nick Ashton and Tommy Karikari. We have been in different email chains discussing a lot of cool studies we will do together with you. And we have been so impressed by the work you are planning and these types of longitudinal studies you are planning with different types of interventions and things like that. This will be, we are so happy that we can get involved in this. And our hope in this collaboration is that we will then contribute these types of biomarker data that Kaj showed you on blood samples from the people who are involved in your studies. And hopefully we'll then together with you be able to see if any of these interventions could change Alzheimer's in the brain. I mean, that would be such a wonderful thing to see. So we have Nick Ashton, Tommy Karkar, and this is Juan Lantero, who has developed a lot of different tau assays in our team. So here you see where who will get your samples eventually. And this is a busy slide, of course, but here you see Kai drew this picture a long time ago, but here you see a neuron. So this is the neuronal soma, the body of the neuron, and then a typical neuron sends out one long extension like this that then connects to other neurons. And it's believed that one neuron connects to approximately 5,000 other neurons in these complex networks that make us think and act and do things. And here, tau is this protein that is then part of the skeletal system of these thin extensions. And it is believed, there are data indicating that tau is part of the cortical, it's highly expressed in thin, unmyelinated, a little bit shorter extensions, axons. Neurofilament light is also a skeletal protein that builds up the skeleton of the large part of the axons here, the large myelinated axons. And in our lab, we have tried to develop targeted assays for these connections, the synapses with different proteins here, tau, neurofilament light, phosphorylated tau, as Kai showed so much data from, and also this type of antennas, the dendrites, neurogranin is another protein. And then we have other cell types of the brain, astrocytes and microglia, where there are different proteins that are released from these cells when they get activated by inflammation or injury or whatever one can think of. And then we have the amyloid plaques here, where we can measure beta amyloid proteins. And as Kai said, we started out with measuring this in cerebrospinal fluid, where you have to do a lumbar puncture. In the back, lower back, you put in a long needle. It is in Sweden, we love to do this procedure, and no one is worried about it. But in some parts of the world, a lumbar puncture is regarded as a bit invasive and perhaps a little bit scary. But we have had a long tradition of doing it. But lately, then we have the possibility of measuring these proteins in blood, thanks to these technological breakthroughs that Kai outlined. But one could also, when looking at this figure, there are some problematic biomarkers that will be hard to translate into blood tests, where we would like to continue to measure them in cerebrospinal fluid or with molecular imaging technologies. But our expertise is the fluids, and that actually relates to the astrocytic and microglial biomarkers, because they are also expressed in cells that are outside the brain. So the blood level is mostly reflecting immune cells of the blood and other non-cerebral tissues, with one exception, and that's a protein called GFAP, which is a promising blood biomarker for inflammation. I will in these few minutes actually focus now on neurofilament light and longitudinal studies we have done with this marker. And I will start with some cerebrospinal fluid investigations, and then show how the blood test for neurofilament light can be used to see almost the same things as we have been able to see in the CSF studies. So I will focus on biomarker dynamics. And the reason for why I will focus on this is actually, again, to think about your intervention studies and your clinical cohorts, where I would like to see biomarker changes reflective of improved brain health, basically, depending on the interventions you will do. This is an old study Kai and I did many years ago on cerebrospinal fluid, where we collected cerebrospinal fluid to measure this injury marker following voluntary exposure to brain damage, that is boxing. So amateur boxing in Sweden, and then these were quite good boxers, and they enrolled in this study, where they came 1 week after their fight and gave a CSF sample to us. Here you see control levels. These are friends of the boxers, and here you see neurofilament light concentrations in the cerebrospinal fluid after they had participated in a boxing bout. And then you see how neurofilament light levels increase in these individuals. And these are actually the ones with the strongest increase. They were the ones who took more hits to their heads. So these actually, if we dichotomize the results at looking at those who received more than 15 punches to their head, these are the ones. No one was knocked out. It was amateur boxing, but this one was a little bit groggy. And this is after the summer rest of the boxing. So the levels are increased after the brain injury, and then they normalize. And this is 3 months of rest from boxing. So neurofilament light looks like this. And it basically, I mean, to return to what we discussed before, this reflects the long extensions between the neurons that they break apart and release their content of neurofilament light into the fluid surrounding the neurons, and also it appears in the lumbar CSF, and we now know that it's also possible to see in the blood. Here is GFAP, the inflammatory marker, and this is Tau, as Kaj mentioned. So increased levels after the bout, and then this is a knocked out boxer. So this boxer received a knockout punch, and then I came 10 days after and gave a CSF sample with very abnormal neurofilament light levels, reflecting the axonal injury he had experienced at the knockout 10 days before. And then this boxer wanted to see that his levels normalized, and he didn't want to box until they were normal. And here you can see the biomarker change over time. So 2 months later, like this, no new injuries, and then it takes, in him it actually took 8 months for the biomarker to normalize completely. So it's not a very fast marker. It's a marker that reflects axonal injury, and then it goes away slowly. This is CSF neurofilament light levels in a treatable neurological disease, multiple sclerosis. So in multiple sclerosis, we have a lot of effective treatments. So here you see multiple sclerosis patients before and after 6 months of treatment, 6 to 12 months of treatment with natalizumab, which is a very effective anti-inflammatory drug that really actually stops the disease process in many of the multiple sclerosis patients. And here you see how beautifully the biomarker normalizes. And actually the levels are similar as in healthy controls. So we have been quite fascinated by this, and we would of course like to be able to see this type of biomarker change also in the neurodegenerative studies. Perhaps you heard yesterday that the antisense oligonucleotide treatment against Huntington has been cancelled, and the NFL change in that study was actually that the drug did not normalize neurofilament light levels. In fact, they saw a bump, a slight increase in neurofilament light, which probably didn't reflect anything good. So neurofilament light in injury and neuroinflammation reflects the axonal injury and neuronal injury process, and you can see that the treatment can normalize the biomarker, which we think is important. And this is NFL data in another neurodegenerative disease, which actually is treatable. And this is the rare pediatric childhood neurodegenerative disease, spinal muscular atrophy, where kids have a mutation in the gene encoding a protein called survival of motor neurons. And then they get a spinal cord atrophy, which will kill them. So it's a severe disease. Some companies have developed, researchers and companies have developed a beautiful type of treatment where an antisense oligonucleotide can be injected in the lumbar sac, and this oligonucleotide will stabilize the mRNA, the template for the protein, and make the protein come back in the spinal cord. And what happens then is that after a few treatments, the NFL levels normalize dramatically like this. On the x-axis here, you see the children's age, and the crosses here are normal children, or children without neurodegeneration. They had suspected brain inflammation, but that turned out to be negative. So these are the control levels. But you see here that a neurodegenerative disease where you have an effective and clever treatment can normalize the biomarker. The biomarker wasn't needed here because the clinicians could see that the kids did better and survived, and also retained motor functions. But for us in the lab, it was important to see this result. And I think it's a fascinating result. In another condition here, here is a completely different type of condition that will impact brain health, and that is HIV infection. So if you get HIV, the virus will rapidly cross the blood-brain barrier and establish itself in oligodendrocytes and microglia, that are two cell types in the brain. And then you actually get a process where the myelin sheath around the long extensions of the axons are destroyed, and then you get neurofilament light increase. So here is a study, a longitudinal study we did together with our colleagues in the infectious disease department. This is a primary HIV infection with flu-like symptoms and high RNA levels in both blood and CSF. And the CD4 cell count, the immune cell count, is normal, and then you do nothing. But then it went down like this, and then you start with antiretroviral treatment, anti-HIV medication. And then the CD4 cell counts go up, and the RNA levels of the virus go down like this. The neurofilament light injury marker looked like this in this study. It slowly increased, and then it went down again. This was the gray zone here indicates normal values. So this was not particularly dramatic, but still a bit interesting, we thought. This is another HIV infection with low CD4 cell counts from the start, high HIV levels, and then CSF NFL concentration that is elevated. So indicating that there is ongoing neuroaxonal injury in the brain. And then treatment was initiated, and the biomarker dropped down like this and became normal. So the biomarker can detect a neuroaxonal injury for different reasons and normalize in response to treatment. This is HIV-associated dementia. So here HIV has caused brain damage so that the patient is demented. And then in this case, this was eventually discovered with very abnormal HIV RNA levels or high, and then low CD4 cell counts, very high CSF neurofilament light concentration, indicating ongoing brain injury. And then in response to treatment, the biomarker slowly normalized, and the neurocognitive, the dementia went away. So it was a reversible dementia in this case, and reflected by the CSF NFL levels. Now we can measure in blood. So when we developed the very sensitive assay for neurofilament light, we started out with severe traumatic brain injury patients, where you have on the x-axis days after the brain trauma, the traumatic brain injury, and a massive release of neurofilament light levels into the bloodstream. And the higher the levels, the more brain injury and the worse outcome. So this looked from a biomarker perspective promising, this result. This is another study where we looked at, we used actually a clinical trial up in northern Sweden, in Umeå, where the idea was to treat progressive multiple sclerosis with a drug called rituximab, but to deliver it inside the brain, in the ventricles of the brain through an intraventricular catheter. And when you place the catheter through the frontal lobe, you will induce a small brain injury. It's very minor, and will not affect the patient. But this gives this type of blip in the CSF neurofilament light concentration. And this blip you could see here was completely reflected in the blood with a similar dynamics. So we think that what we could see before in CSF, we can now see in this simple blood sample. And that is why we are so interested in measuring this protein, and also, of course, the AD biomarkers in the studies you are doing. Here is in another brain condition, less severe trauma, but concussion in ice hockey. So ice hockey is a big thing in Sweden as it is in the US. And concussion is a big problem. I mean, that's really, if you're a professional ice hockey player, you can count on getting at least 2 concussions per year in your profession, which is, I'm happy not having that profession. What we did was that we collected blood samples 1 hour, 12 hours, 36 hours, and 144 hours after the concussion. And then when the ice hockey players could return to play, which was approximately 1 week after the concussion. That is probably a bit too short. And then we analyzed the samples. And this was the result, which to us was a bit disappointing in the start because it was such a messy picture, but we now think this makes sense. So here you see that most concussions in the ice hockey players did not cause a neurofilament light increase. And to us, this indicates that most of the concussions did not cause a detectable axonal injury or diffuse axonal injury process, but in some players take off in their injury marker like this. And these are probably, but we don't know this, but we think these are the players who should rest longer and try to avoid additional concussions. So around 10% of the concussions in these studies we have done in ice hockey have clear biomarker evidence of a frank brain injury, whereas perhaps these ones are a little bit less severe. So we are trying to work a little bit more with this and see if we can figure this out better. But to me, it sort of makes sense when looking at if all concussions gave a lot of axonal injury, perhaps one would see even more ice hockey players having problems. But brain trauma is definitely something one should examine in the cohorts, of course, and we think neurofilament light can be used to detect such things. Anesthesia and surgery is another life course event that will happen to many people, and that is associated with some brain biomarker evidence of brain injury when looking in the blood. So here you see hours after general anesthesia in relation to different surgery types. The NFL data are increasing a little bit like this. It's not dramatic, but NFL is a slow marker, as I said, also with a maximum after several days. And here you see tau instead, which is a more rapid biomarker. So if you have general anesthesia, you get this type of tau signal in the blood. And it's quite dramatic when looking at the percentages here. So it's most likely not that good to go into this type of most likely it's a quite severe brain challenge to undergo surgery with general anesthesia, and that those of you who are clinicians will know about delirium and cognitive worsening after surgery. These data were a bit hard for us to interpret, but this study was to us much more exciting and easier to interpret. So this we did together with Polish researchers. These are data from patients who undergo cardiac surgery, and around half of them did cardiac surgery with a beating heart with normal circulation, whereas the other half did the surgery with extracorporeal blood circulation in a heart and lung machine with laminar blood flow. And here you see the tau data on those who had a beating heart. And here you see those who were on pump. And you see this type of strong difference between the two groups. A lot of things could happen here. These are hours during the surgery, and this is the day 7 time point. But here you see at day 7 for neurofilament light that there you see a strong increase. And actually the anesthesiologists, they have coined a term called pump head, which is, I didn't know before this study was published, but this is the knowledge that this type of cardiac surgery with extracorporeal circulation is associated with brain dysfunction, delirium, and other things. And the biomarker can help identify that. Here you see also mild traumatic brain injury data, moderate to severe brain injury data looking at dynamics. And most of the injury markers we are used to using, they have a downward slope. They are increased early and go down with time, but neurofilament light is the slower dynamic. So it looks like it peaks around 2 to 3 weeks after brain injury. I think I skipped this so we can discuss. This is a neurodegeneration. Here is Huntington disease. CSF and plasma neurofilament light levels in patients who carry a mutation that will cause them Huntington disease. And the bluer the color, the darker the color, I should say, the more severe genetic mutation. And you can see how people shoot off in their neurofilament light levels like this when they enter the neurodegenerative phase. So we think that neurofilament light is a marker for onset of neurodegeneration. And then you see here familial Alzheimer's disease data. Mutation carriers who are entering the neurodegenerative phase, and the blue lines represent non-carriers, and you can see that the biomarker is stable in those. And this is ADNI data. I think I will skip so we can discuss also a bit because I see the time is running out. This is a slide showing blood neurofilament light results when you treat multiple sclerosis with natalizumab, and how this type of normalization happens in the patients, which is something we are looking for in neurodegenerative diseases. So with this, I think if we summarize Kaj and my talk, we have fluid biomarkers for AD pathology and they also work in blood. They are dynamic, which is important. Repeated measures in clinical trials, cohort studies, and in intervention studies that you are planning will help us understand the biomarkers better. And it will also tell if the interventions actually do something to brain health and underlying pathology. So these studies will be super exciting that we will do with you. I think also in clinical trials, it will be important to know about longitudinal stability of the biomarker concentrations. And the blood biomarkers will make it much easier to do longitudinal studies, population-based and other types of studies where we won't have to do repeated expensive imaging measurements or CSF collections, but simply do the blood tests. And perhaps also use imaging at the start and end of the study or something like that, but have more dense sample collection for the blood biomarkers. And with that, I close here, and so that we have some time to discuss also. Thank you very much.
Morten, I have questions.
Sure. Why don't you hold on one second, Laszlo? I'll call you 1 second. So Henrik, that was great. And Kaj, I guess I can't see both of you at once because we've got 51 people here. But thank you both for these two different perspectives on that. I did want to ask one question because you were showing in in your neurofilament light where it goes up, it goes down, depending on injury and recovery. In any of the phosphorylated tau, have there been any, particularly with the CSF, which has been out for a while, they've been showing it progressing over time. There have been longitudinal studies showing it getting worse. Has there been any interventional studies in the CSF, or even the PET imaging, which have shown these AD biomarkers going down? Has there ever been a negative slope on any AD biomarker longitudinal graph?
Okay. I can start maybe. I mean, there are some early data from the Alzheimer clinical trials with A-beta antibodies. So it's quite small number of cases, but there was reported a decrease in Phospho Tau, for example, in cerebrospinal fluid with treatment using either aducanumab or the other antibody ACIs BAN2401. There was also, I think in the ACI study, a reduction in neurofilament light and the synaptic protein neurogranin. But as said, it's a rather limited number of patients and preliminary data.
And we could add also that the blood biomarker, of course, all these interrupted or closed trials, they have a vast amount of samples in freezers. And one would so very much like to see plasma phospho tau and neurofilament light from those sample collections. But still, we do not, we haven't seen any, at least I have not seen any data being presented on that. So we always tease the pharma company representatives we have been working with and ask them to do this, of course. They can do it. And it would be so informative to be able to look at, for example, Phospho Tau 181, 217, 231, GFAP, neurofilament light in large numbers of people who have received these different doses. And so is there a biomarker sign or not? But we haven't seen it yet.
Are they resistant to? I mean, they have these blood banks. They bank it on these studies, but no one's gone back retrospectively to.
No, and I think they are waiting now to see what happens with aducanumab. And they, at the moment, my feeling is that they don't want more data.
I see. I was going to ask why they're not jumping it, because they're afraid that the data will be negative and that will kind of be.
Yeah, or perhaps an unexpected result. It's hard to tell, actually. But Maria Carillo at Alzheimer Association, and some other some other organizations are really trying to form consortia and there are also pharma people who try to make the samples available.
Great. Let me just say, Laszlo, you had a question. Did you want to?
Right. So it's a beautiful progress in the diagnosis, but let me ask you, are you planning, or you had already data when these blood markers are correlated, either the cognitive domains, so memory, language, visual, spatial, or structural changes which are well known in the hippocampus or basal forebrain, because the specificity, it's an issue. And the second, we still don't know what is the time course of the structural damage. It's a second one, which is probably...
Well, Laszlo, why don't we do one question? Laszlo, let's just do one question.
The question is, if you correlate with structural or cognitive domain, and how much you can predict, MCI has a stable and a so-called converter group. So one which remain stable in 10 years, a certain part convert to Alzheimer. So can the blood biomarker used for predicting?
Mm-hmm.
Should we start with, if I start with this neurofilament light biomarker, it is really, it is actually predictive of future brain changes in longitudinal magnetic resonance imaging studies. So the higher the neurofilament light level in an MCI population, the more loss of brain tissue, the larger the ventricles get. But it's not, and that's very important to remember, it's not a disease-specific biomarker. It will predict all cause neurodegeneration, sort of. But then the longitudinal studies on Phospho Tau and the beta 42 look the predictive studies there look much more AD specific. If you would like to comment on that, Kaj.
Thanks.
Yeah, I think it's similar for the Tau and Phospho Tau, meaning that if you have, for example, lower beta 42 to 40 ratio in MCI cases or cognitively unimpaired, there are some data showing that this is the group that will show cognitive deterioration. Absolutely, yes.
Great. Okay, why don't we? I'd love to get some of our graduate students, especially any of the graduate students from any of the programs. Is there anyone who wants to jump up with a question from either the BNS students, or I know we have a number of MPH students. No, none of our graduate students want to jump in and ask a question. Malta, you always have good questions. I'm going to call on you. Is there anything you wanted to ask? You're still there? No. Okay.
No, I'm here. I just don't have a good question.
Oh, that's surprising. Is there anyone else who wants to ask some questions, particularly some of our distal visitors who've come in from, we have a huge representation from the YASA lab and the Irvine group, except for the eponymous YASA, who I don't think is here, but a huge number of other people there. From the Irvine group, can we, anything you'd like to ask or? There. I don't seem to be getting any questions. Shirin, there you are. There's one of our BNS students.
Hi, so I'm just wondering, like, what's the contribution of the enteric nervous system to this kind of blood markers as compared to the central nervous system?
That's a great question. It's a problem for us. We really don't like the enteric nervous system because it contains neurofilament light. So that's another. So peripheral nerves and central nerves both contain neurofilament light. And we have seen in, for example, Charcot Marie Tooth, which is a peripheral neuropathy, that the levels are a little bit increased in plasma. So we are not certain, but in most cases, there is a nice correlation between CSF and blood levels. and the CSF concentration is much higher than the blood concentration. So we think that most of the plasma neurofilament light concentration reflects central neurofilament light release, but we can't be completely certain. And for example, in the COVID studies we have done, if you get moderate to severe COVID, you get the neurofilament light increase. And we think that it looks like it can predict some of the neurological problems that may occur in COVID, but there is also a neurophysiological evidence that part of that signal seems to come from peripheral nerves. And the way we have tried to address this is to actually try to develop some additional assays, because there is one CNS selective or enriched form of neurofilament, and that protein is called alpha internexin. And we are trying to make an assay for that. Then there is a neurofilament which is highly enriched in peripheral nerves and not at all expressed intracranially. And that protein is called peripherin. And there I also have a lab in London. There we have had one a graduate student who has made an assay that works reasonably well for peripherin. So there is, so we hope to be able to actually address this in the future, but we have no data yet.
Okay.
Thank you so much.
Thanks. Any other questions? We'd love to. Anyone else want to? I can only see half the people at once, so people should just jump in and raise their voice, if not their hands, if they want to ask a question.
This is Denise Kirk from Kessler Foundation.
Great.
Hi, thanks for really great talks. I'm new in getting into blood biomarkers in my research, and I have probably what is a very, very basic question, but I was wondering if there is a difference in sensitivity to picking up all these different markers, whether they're the AD-specific biomarkers or whether they're NFL, in whether you select to use plasma or serum.
That's a very good question also, and it differs a little between the biomarkers. So traditionally, beta amyloid has been measured in plasma. So it seems to work better in plasma. For NFL, it doesn't really matter, and I think it's the same for Phospho Tau in principle. And the reason for this is not really known in detail.
So if you were to choose a method that would be the best option for measuring a number of biomarkers, say you wanted to measure AD-specific biomarkers as well as inflammatory biomarkers, would it be better then to choose plasma? I noticed that in much of the slides, you were referencing studies where they used plasma.
Yeah, it looks like that, I think. But I think you have to test it for each and every biomarker separately.
And when you biobank, when you meet all these study participants and get blood samples, the advice would be to biobank some serum, some plasma, some whole blood, perhaps, so that because then you future-proof your biobank. Because there might be a future biomarker that comes up that you only can measure in serum, potentially, or vice versa, or the other way around. So for biobanks, one should probably collect different biofluids, but most studies use EDTA plasma, at least in this field. And most biomarkers, I'm thinking about what can't be measured in EDTA plasma at the moment, but yeah, most, there is S100B, for example, is a protein, but that's not a very interesting protein either. So.
Diana is one of the undergraduates in my lab. You want to?
Yes. Thank you so much for your presentation. I just have a basic question. So you said that p-tau 217 shows very high performance to identify Alzheimer's disease. My question is how it differs with p-tau 181. So we have two different blood biomarkers. So I guess no.
Absolutely, and I think the problem today is that there are different assays, analytical methods for p-tau 181 and 217 and 231, and then you have evaluated these methods in different cohorts, and then you compare data. So I tried a little bit to say this between the lines, that for sure in the JAMA paper, Phospho Tau 217 worked very well. But then if you go to other papers with neuropathology, for example, 217 was even better, and 231 was one step better again. So I think we need to await the studies where you have similar assays for these three phosphoepitopes and see if there really is a difference. And we tried that to some degree in CSF, and there they, as I showed on one slide, and they were really similar and correlated tightly. And at least in my eyes, I can't find a good explanation why it would be different in plasma than in cerebrospinal fluid. So my guess is that they will correlate tightly, and the best way forward is maybe to have different highly validated assays for these three variants of Phospho Tau that compete against each other.
Could I ask, what do you think about this idea with that the 217 phospho form is more CNS, perhaps only happens in neurons that are ill?
Are you thinking of the Barthélemy paper?
Exactly. Yeah, I thought it was fascinating.
To be honest, I've read that in detail, and I think the data supporting
Swedish people.
I love
the data supporting this idea was that the fold change, so the degree of increase, was higher for 217 than for 181 in that specific cohort. So again, I think we need to evaluate it again in different cohorts using similar assays.
So actually, let me just ask two questions about these different assays. One is, do you think any of them, obviously, once you're in the blood, you're far away from the brain, but are there any assays that might differentiate between what is kicked off from, say, the medial temporal lobe versus brainstem versus cortex? And the second question is there any potential that these different assays may be differentially sensitive at different phases? So someone who's 15, 20 years out, maybe this assay is really going to show sensitivity, but once you get up to sort of, getting close to MCI, maybe that's no longer useful because it's plateaued and hit a ceiling. But there are other measures which might be much more useful if you're in that range of MCI versus medium. So is there anything that metric might differentiate?
That's a good question, and it has been discussed specifically this, whether one variant is more sensitive early on and the other is better in the late stages, and there are some indications for this for sure, but at the same time I think we need more studies again to be certain whether there are this type of difference.
We need you and your team, Mark. Because actually it's so interesting, because the small differences that have been seen between the different Phospho Tau biomarkers, we thought for a while that Phospho Tau 231 perhaps was a little bit earlier, but then actually we have seen another study where it's not that clear, and it will probably be a few years where we eventually will figure this out together with you have the cohorts.
Well, I mean, actually, going back to one of the things that Laszlo had said, he was asking about specificity to cognitive function and hippocampal dynamics and so forth. So obviously, we're going to be, measuring all of those things. We're going to be measuring, a broad range of cognitive functioning, including sensitive measures of entorhinal and hippocampal function. We'll be looking at these dynamic resting state connectivity measures that we've been using that give you something not only about the structure, but about the sort of the flexibility of these connectivities. So we're particularly excited. Those very more sort of refined cognitive measures and those more refined neural connectivity measures, I think, are going to be very exciting to sort of link them up to these different markers. Let's see whether they're at least will have sensitive cognitive and sensitive, neural network dynamic measures that we can link up.
One interesting thing is really this interplay between degeneration and plasticity. I mean, that's super hard for us to address in the fluids. But but there are some biomarker. I mean, the synaptic markers potentially could reflect some plasticity changes, and there are hypotheses that, for example, the 181 phosphorylation might be also happening in normal synaptic and axonal turnover, and perhaps some other phosphoform is more neurodegenerative and so. But with your measures, perhaps we can get some hints on this.
Yeah, no, it'll be fascinating to see whether there's any differentiation among these measures when we start actually getting at much more sort of refined cognitive or neural network functioning.
Paul Rapp, if I might, question for Professor Zetterberg. I'd like to confirm my understanding of one of the diagrams you showed. It was neurofilament light following concussion. That within that participant cohort, there was a subset where NFL actually was increasing rather than resolving as a function of time. Am I? Okay. Exactly. I'll follow up, sir, with email on this, because we are actually conducting studies and taking blood samples of people who experiencing multiple non-catastrophic concussions. Typically, individuals who are firing rocket-propelled grenades from a, actually, the weapon is named after a king of Sweden. It's the Carl Gustaf rocket launcher.
Yeah.
So, I mean, there must be a connection here. But one of our big concerns, as you were showing about return to play decisions, we have return to duty decisions. And if we can find a blood marker which says, this chap can go back to work, this one needs to stand down, this will be extremely important to us. So as I say, first of all, did I read that diagram correctly, that there was a subset of, and because we do know from animal studies that diffuse axonal injury can actually propagate, can progress for, in the case of rat studies, for up to a year after injury, which is a third of the animal's life. So the idea of progressive diffuse axonal injury is something which we need to investigate.
Yeah.
And I'm
very encouraged by
what I saw this morning.
Yeah, this is where we think neurofilament light works as a biomarker. We will also send you a study we did together with the Swedish military armed forces on Carl Gustaf exposure, firing of heavy weapons, because there we did actually a CCF study. I'll send it.
Please, I'll make contact via email. So.
Yeah, that's great.
Okay, I think we've kept you guys. It's probably dinner time in Gothenburg, so you guys are probably getting hungry. But I just want to thank you guys so much for coming and joining us and sharing a lot of your exciting data and entertaining our questions. And of course, most of all, thank you for your enthusiasm in eagerly awaiting the blood samples, which we now have N of 1. And when we begin over the next few weeks, as our lab reopens, we'll hope to bring considerably higher than N is 1. So we send you a batch and then talk more. So I agree that right now we're sending the longitudinal data, and Bryce is excited to look at the sleep correlates of a lot of these things. I think that's a whole other domain whereby we can look at these different measures and ask, are any of them linking up particularly to the microstructure of sleep disorders, to REM sleep or slow wave sleep or spindles, as we look at any of these things that are being disrupted in aging and Alzheimer's, to ask how they're correlated. And as you point out, I think, although we're at the moment, we're in a pause between our exercise intervention studies, which ended fortunately just before COVID, and others which will start up, perhaps in the fall or later, that the possibility that we could use some of these to actually track, to ask whether or not we know exercise, cardiovascular exercise is neuroprotective. We know that it improves neural functioning, but we don't know whether it's doing anything at the pathology level. And I think it's a fascinating question where sort of any answer is interesting. Obviously, if we see the pathology going backwards, then that's, tremendously exciting. But on the other hand, if we see that all of these neural and cognitive improvements are in the absence of any change in the trajectory of the pathology, then that's telling us that a lot of these lifestyle and exercise is sort of compensatory. And that in itself is important, and useful to know, because of course, in the end, we don't really care about the pathology. We care about our thinking and functioning in the world, and how we get there is exciting to know. So anyway, we're all very excited to work with you, excited to hear about your work. And I will thank you all, and thank all, well, we're back down, all of you who came and joined us from California and St. Louis, and of course from Sweden. Take care. Bye-bye.
Thank you all. Bye-bye.
Thank you so much. Take care.
Thank you.